Welcome to the video lecture for Unit 5, Module 11.2. In this one, we're going to start a small series on some of the anabolic and catabolic reactions and get into a little bit of biochemistry. Specifically in this one, we're going to talk about carbohydrate metabolism. As always, here are your learning objectives. Be sure to reference these when studying for both your lecture quiz as well as the unit exam. Let's go ahead and get started. Most cells generate ATP and other high-energy compounds by breaking down carbohydrates, especially glucose, and we can summarize the complete reaction as follows. You have glucose plus oxygen will release carbon dioxide and water. Most people always remember two things when it comes to biology. One is that the mitochondria is the powerhouse of the cell, which we're definitely going to talk about in this module. And two is usually, or for those of us that have taken any sort of advanced or post-secondary scientific course, is the chemical structure of glucose, which is C6H12O6, and you can see that reaction on the page here. The breakdown takes place in a series of small steps. Several of these steps release sufficient energy to convert ADP to ATP. The most complete metabolism of one molecule of glucose provides a typical body cell with a net gain of 36 ATP molecules. Most ATP production takes place inside the mitochondria, but the first steps take place inside the cytosol. In 168, we introduced the process of glycolysis, which breaks down glucose into cytosol into smaller molecules that the mitochondria can absorb and utilize. These reactions are said to be anaerobic because glycolysis does not require oxygen. The subsequent reactions take place in the mitochondria. These reactions use oxygen and are considered aerobic. The mitochondrial activity responsible for ATP production is called aerobic metabolism, also known as cellular respiration. Glycolysis is the breakdown of glucose to pyruvic acid. In this process, a series of enzymatic steps breaks down the 6-carbon glucose molecule, remember C6H12O6, into two 3-carbon molecules of pyruvic acid, or CH3COCOOH. At the normal pH inside the cells, each pyruvic acid molecule loses a hydrogen ion and exists as the negatively charged ion CH3COCOO-. This ionized form is called pyruvate. Glycolysis requires 1. Glucose molecules, 2. Appropriate cytosolic enzymes, 3. ATP and ADP, 4. Inorganic phosphates, and 5. NAD, or nicotinamide adenine dinucleotide, which is a coenzyme that removes hydrogen atoms during one of the enzymatic reactions. Recall from 168 that coenzymes are organic molecules that are essential to form enzyme function. If any of these participants is missing, glycolysis cannot take place. Glycolysis begins when an enzyme phosphorylates, that is, it attaches a phosphate group to the last or the 6th carbon atom of a glucose molecule, creating glucose 6-phosphate. This step costs the cell 1 ATP molecule. It has two important results. 1, it traps the glucose molecule within the cell because phosphorylated glucose cannot cross the plasma membrane. And 2, it prepares the glucose molecule for further biochemical reactions. A second phosphorylation takes place in the cytosol before the 6-carbon chain is broken down into two 3-carbon fragments. The energy benefits begin to appear as these fragments are converted to pyruvate. Two of the steps release enough energy to generate ATP from ADP and inorganic phosphate, or PO4. In addition, 2 molecules of NAD are converted to NADH. The net reaction of glycolysis looks like the reaction that you see on the page, which is glucose plus 2 NAD plus 2 ATP plus 2P is equal to 2 pyruvate plus 2 NADH plus 6 ATP. This reaction sequence is anaerobic, or without oxygen, and provides the cell with a net gain of 2 molecules of ATP for each glucose molecule converted to 2 pyruvate molecules. A few highly specialized cells, such as red blood cells, lack mitochondria and derive all of their ATP through glycolysis. Skeletal muscle fibers rely on glycolysis for ATP production during the periods of active contraction. Most cells can survive for brief periods using the ATP provided by glycolysis alone. However, when oxygen is readily available, mitochondrial activity provides most of the ATP that the cell needs. For the cell, glycolysis yields an immediate net gain of 2 ATP molecules for each glucose molecule that it breaks down. However, a great deal of additional energy is still stored in the chemical bonds of pyruvate. The cell's ability to capture that energy depends on the availability of oxygen. If oxygen supplies are adequate, mitochondria absorb the pyruvate molecules and break them down. As you'll see in the next session, the hydrogen atoms of each pyruvate molecule are removed by coenzymes and are ultimately the source of most of the cell's energy gain. The carbon and oxygen atoms are removed and released as carbon dioxide in a process called decarboxylation. Recall that a double membrane surrounds each mitochondrion. The outer membrane contains large pores that are permeable to ions and small organic molecules such as pyruvate. Ions and molecules easily enter the intermembrane space, separating the outer membrane from the inner membrane. The inner membrane contains a carrier protein that moves pyruvate into the mitochondrial matrix, where it's broken down through the citric acid cycle. In the mitochondrion, a pyruvate molecule participates in a complex reaction involving NAD and other coenzymes called coenzyme A or CoA. This reaction yields one molecule of carbon dioxide, one of NADH, and one of acetyl-CoA, which is a 2-carbon acetyl group bound to a coenzyme. This sets the stage for a sequence of enzymatic reactions called the citric acid cycle. It's also known as the tricarboxylic acid cycle, or the TCA cycle, and the Krebs cycle. Because citric acid is the first substrate of the cycle, we'll use citric acid cycle as the preferred term. The function of the citric acid cycle is to remove hydrogen atoms from organic molecules and transfer them to coenzymes. At the start of the citric acid cycle, the 2-carbon acetyl group carried by CoA is transferred to a 4-carbon oxaloacylic acid molecule to make the 6-carbon compound citric acid. Coenzyme A is released intact and can thus bind to another acetyl group. A complete revolution or turn of the citric acid cycle removes 2 carbon atoms, regenerating the 4-carbon chain, and this is why the reaction sequence is called a cycle. We can summarize the fate of the atoms in the acetyl group as follows. The 2 carbon atoms are removed in enzymatic reactions that incorporate 4 oxygen atoms and form 2 molecules of carbon dioxide, which is a waste product. The hydrogen atoms are removed by the coenzyme NAD or a related coenzyme called FAD, or flavin adenine dinucleotide. Several of the steps in a turn of the citric acid cycle involve more than one reaction and require more than one enzyme. Water molecules are tied up in 2 of these steps, and we can summarize that entire reaction sequence as you can see on the screen there. The only immediate energy benefit of one turn of the citric acid cycle is the formation of a single molecule of GTP, or guanosine triphosphate, from GDP, or guanosine diphosphate, and phosphate. In practical terms, GTP is the equivalent of ATP because GTP readily transfers a phosphate group to ADP, producing ATP. That reaction's on the screen as well, which is GTP plus ADP will equal GDP plus ATP. The formation of GTP from DDP in the citric acid cycle is an example of substrate-level phosphorylation. In this process, an enzyme uses the energy released by a chemical reaction to transfer a phosphate group to a suitable acceptor molecule. GTP is formed in the citric acid cycle, but many reaction pathways in the cytosol phosphorylate ADP and form ATP directly. For example, the ATP produced during glycolysis is generated through the substrate-level phosphorylation. Normally, however, substrate-level phosphorylation provides a relatively small amount of energy compared with oxidative phosphorylation, which we'll discuss next. Oxidative phosphorylation is the generation of ATP within the mitochondria in a reaction sequence that requires coenzymes and consumes oxygen. The process produces more than 90% of the ATP used by body cells. The key reactions take place in the electron transport system, or the ETS, which is a series of integral and peripheral proteins in the inner mitochondrial membrane. The basis of oxidative phosphorylation is the formation of water, which is a very simple reaction. 2 hydrogen atoms plus oxygen equals 2H2O. Cells can easily obtain the ingredients for this reaction. Hydrogen is a component of all organic molecules, and oxygen is an atmospheric gas. The only problem is that the reaction releases a tremendous amount of energy all at once. In this fact, this reaction releases so much energy that it's used to launch space shuttles into orbit. Cells cannot handle energy explosions. Instead, energy release must be gradual, as it is in oxidative phosphorylation. This powerful reaction proceeds in a series of small, enzymatically controlled steps. Under these controlled conditions, energy can be captured safely and ATP generated. The enzymatic steps of oxidative phosphorylation involve oxidation and reduction. There are different types of oxidation and reduction reactions, but for our purposes, the most important are those involving the transfer of electrons. The loss of electrons is a form of oxidation. The gain of electrons is a form of reduction. One way to easily remember these things is that when you reduce a molecule, it is becoming more negative. The addition of more electrons to the system is going to make the system overall more negative. So you're therefore reducing the charge of the atom or making it more negative. Thus, it's a reduction reaction. The two reactions are always paired. When electrons pass from one molecule to another, the electron donor is oxidized and the electron recipient is reduced. Oxidation and reduction are important because electrons carry chemical energy. In a typical oxidation-reduction reaction, or a redox reaction, the reduced molecules gain energy at the expense of the oxidized molecule. In such an exchange, the reduced molecule does not gain all the energy released by the oxidized molecule. Some energy is always going to be released as heat in almost every single chemical reaction that you see. Most chemical reactions are not 100% energy efficient. The remaining energy may be used to do physical or chemical work, such as forming ATP. By passing electrons through a series of redox reactions, cells can capture and use much of the energy that is released as water is formed. Water is formed as these electrons ultimately combine with hydrogen ions and oxygen atoms. Coenzymes play a key role in this process. A coenzyme acts as an intermediary that accepts electrons from one molecule and transfers them to another molecule. In the citric acid cycle, the coenzymes NAD and FAD remove hydrogen atoms from organic molecules. Each hydrogen atom consists of an electron and a proton. Thus, when a coenzyme accepts hydrogen atoms, the coenzyme is reduced and gains energy. The donor molecule loses electron and energy as it gives up its hydrogen atoms. Note that NADH and FADH2 are the reduced forms of NAD and FAD, and then they transfer their newly acquired hydrogen atoms to other coenzymes. The protons are subsequently released. The electrons, which carry the chemical energy, enter a sequence of oxidation-reduction reactions known as the electron transport system. This sequence ends with the transfer of electrons to oxygen and the formation of a water molecule. At several steps along the redox sequence, enough energy is released to support the synthesis of ATP from ADP. Now let's consider that reaction sequence in greater detail. The coenzyme FAD accepts 2 hydrogen atoms from the citric acid cycle. In doing so, FAD gains 2 electrons, forming FADH2. The oxidized form of the coenzyme NAD has a positive charge. This coenzyme also gains 2 electrons as 2 hydrogen atoms are removed from the donor molecule, resulting in the formation of NADH and the release of a proton. For this reason, the reduced form of NAD is often described as NADH plus H+. The electron transport system, or ETS, or a respiratory chain, is a sequence of proteins called cytochromes. Each cytochrome has 2 parts, a protein and a pigment. The protein is embedded in the inner membrane of a mitochondrion. The protein surrounds the pigment complex, which contains a metal ion, either iron or copper. And we'll consider 4 cytochromes, B, C, A, and A3. First, let's look at the general path taken by the electrons that are captured and delivered by coenzymes. In the first step, a coenzyme strips a pair of hydrogen atoms from a substrate molecule. As we've seen, different coenzymes are used for different substrate molecules. Recall that during glycolysis, which takes place in the cytosol, NAD is reduced to NADH. Within mitochondrion, both NAD and FAD are reduced to the reactions of the citric acid cycle, producing NADH and FADH2 respectively. In step 2, NADH and FADH2 deliver hydrogen atoms to coenzymes embedded in the inner mitochondrial membrane. The electrons carry the energy and the protons that accompany them are released before electrons are transferred to the ETS. The electrons travel to the ETS by one of 2 paths. Which one depends on whether the donor is NADH or FADH2. The path from NADH involves the coenzyme FMN, or flavin mononucleotide. The path from FADH2 proceeds directly to coenzyme Q, or ubiquinone. Both MFN and coenzyme Q are bound to the inner mitochondrial membrane. In step 3, coenzyme Q releases hydrogen ions and passes them and the electrons to cytochrome B. In step 4, electrons are passed along the electron transport system, losing energy in a series of small steps. The sequence is cytochrome B to C to A and then to A3. In step 5, at the end of the ETS, an oxygen atom accepts the electrons and combines with hydrogen ions to form water. Notice that this reaction sequence starts with the removal of 2 hydrogen atoms from a substrate molecule and ends with the formation of water from 2 hydrogen ions and 1 oxygen ion. This is the reaction that we described earlier as releasing too much energy if performed in a single step. However, because the reaction takes place in a series of small steps, the hydrogen and oxygen combine safely rather than explosively. As we noted in 168, concentration gradients across membranes represent a form of potential energy that can be harnessed by the cell. The electron transport system does not produce ATP directly. Instead, it creates the conditions necessary for ATP production. It creates a steep concentration gradient across the inner mitochondrial membrane. The electrons that travel along the ETS release energy as they pass from coenzyme to cytochrome and form cytochrome to cytochrome. The energy released at each of the several steps derives hydrogen ion pumps. These pumps move hydrogen ions from the mitochondrial matrix into the space between the inner and outer mitochondrial membranes. The pump creates a large concentration gradient for hydrogen ions across the inner membrane. This concentration gradient provides the energy to convert ADP to ATP. Despite the concentration gradient, hydrogen ions cannot diffuse into the matrix because they're not lipid soluble. However, hydrogen ion channels in the inner membrane permit hydrogen to diffuse into the matrix. These ion chambers and their attached coupling factors make a protein complex called ATP synthase. The kinetic energy of passing hydrogen ions generates ATP in a process known as chemiosmosis, or chemiosmotic phosphorylation. Hydrogen ions are pumped at 3 places, as 1, FMN reduces coenzyme Q, 2, cytochrome B reduces cytochrome C, and 3, electrons are passed from cytochrome A to cytochrome A3. For each pair of electrons removed from a substrate in the citric acid cycle by NAD, 6 hydrogen ions are pumped across the inner membrane of the mitochondrion and into the intermembrane space. Their re-entry into the matrix provides the energy to generate 3 molecules of ATP. Alternatively, for each pair of electrons removed from a substrate in the citric acid cycle by FAD, 4 hydrogen ions are pumped across the inner membrane and into the intermembrane space. Their re-entry into the matrix provides the energy to generate 2 molecules of ATP. Oxidative phosphorylation is the most important cellular mechanism for generating ATP. In most cases, if oxidative phosphorylation slows or stops, the cell dies. If many cells are affected, the individual may die. Oxidative phosphorylation requires both oxygen and electrons. For this reason, the rate of ATP generation is limited by the availability of either oxygen or electrons. Cells obtain oxygen by diffusion from the extracellular fluid. If the supply of oxygen is cut off, mitochondrial ATP production stops, because reduced cytochrome A3 will have no acceptor for its electrons. With the last reaction stopped, the entire ETS comes to a halt, like cars at a washed-out bridge. Oxidative phosphorylation can no longer take place, and cells quickly succumb to energy starvation. Because neurons have high demand for energy, the brain is one of the first organs to be affected. Hydrogen cyanide gas is sometimes used as a pesticide to kill rats or mice, and in some states, where capital punishment is legal, it's used to execute criminals. The cyanide ion, or CN2, binds to cytochrome A3 and prevents the transfer of electrons to oxygen. As a result, cells die from energy starvation. For most cells, the main method of generation of ATP is the complete reaction pathway that begins with glucose and ends with carbon dioxide and water. During glycolysis, the cell gains a net 2 molecules of ATP for each glucose molecule broken down anaerobically to 2 molecules of pyruvate. 2 molecules of NADH are also produced. In most cells, electrons are passed from NADH to FAD by an intermediate in the intermembrane space, and then to coenzyme Q in the electron transport system. This sequence of events ultimately provides an additional 4 ATP molecules. The citric acid cycle breaks down the 2 pyruvate molecules, transferring hydrogen atoms to NADH and FADH2. These coenzymes provide the electrons to the ETS. Each of the 8 molecules of NADH yields 3 molecules of ATP and 1 water molecule. Each of the 2 FADH2 molecules produces 2 ATP molecules and 1 water molecule. Thus, the shuffling from the citric acid cycle to the ETS yields 28 molecules of ATP. Each of the 2 revolutions of the citric acid cycle required to break down both pyruvate molecules completely yields 1 molecule of ATP by way of GTP. This cycling provides an additional gain of 2 molecules of ATP. So summing that up, for each glucose molecule processed, the cell gains 36 molecules of ATP. 2 from glycolysis, 4 from the NADH generation in glycolysis, 2 from the citric acid cycle by means of GTP, and 28 from the ETS. Cardiac muscle cells and liver cells are able to gain an additional 2 ATP molecules for each glucose broken down. They do so by increasing the energy yield from 2 NADH generated during glycolysis from 4 to 6 ATP molecules. Although the NADH produced during glycolysis does not enter mitochondria in these cells, each NADH molecule passes its electron to an intermediate molecule that can cross the mitochondrial membrane. Inside the matrix, NAD strips electrons from the intermediate molecule, forming NADH. The subsequent transfer of electrons to FMN, coenzyme Q, and the ETS results in the production of 6 ATP molecules, just as if the 2 NADH molecules formed during glycolysis had been generated in the citric acid cycle. Some of the steps in the breakdown of glucose or glycolysis are essentially irreversible. For this reason, cells cannot generate glucose simply by using the same enzymes and reversing the steps of glycolysis. Glycolysis and the production of glucose involve different sets of regulatory enzymes. As a result, the two processes are regulated independently. Some 3-carbon molecules other than pyruvate can be used to synthesize glucose. For this reason, a cell that can generate glucose molecules from other carbohydrates, lactate, glycerol, or some amino acids. However, cells cannot use acetyl-CoA to make glucose. The reason is that the decarboxylation step, or the removal of CO2 between pyruvate and acetyl-CoA, cannot be reversed. Gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors such as lactate, glycerol, or amino acids. Fatty acids and many amino acids cannot be used for gluconeogenesis because their catabolic pathways produce acetyl-CoA. Glucose molecules formed by gluconeogenesis can be used to manufacture other simple sugars, complex carbohydrates, proteoglycans, or nucleic acids. In the liver and in skeletal muscles, glucose molecules are stored as glycogen. Glycogen is an important energy reserve that can be broken down when the cell cannot obtain enough glucose from interstitial fluid. Glycogen molecules are large, but glycogen reserves take up very little space because they form compact, insoluble granules. The formation of glycogen from glucose, known as glycogenesis, is a complex process. It involves several steps and requires the high-energy compound uridine triphosphate, or UTP. The breakdown of glycogen, called glycogenolysis, takes place quickly and involves a single enzymatic step.